Hello, my name is Greg Petsko, and I'm professor of neurology at Harvard Medical School and Brigham and Women's Hospital in Boston. On the 9th of November 1989, when the Berlin Wall fell, I was a professor of chemistry at MIT, and I wasn't working on human diseases at all. Now I'm professor of neurology, a field in which I have not taken so much as a single course. If that doesn't say something about the importance of lifelong learning, I don't know what does. A very big one. My research is aimed at smashing the wall that is amyotrophic lateral sclerosis, or ALS. You may have heard it also called Lou Gehrig's disease. If there's a worse disease, I would never want to see it. Unlike other neurodegenerative diseases, which progressively rob the brain of its ability to think or damage the personality, ALS destroys the motor neurons, which are the nerve cells that connect the brain to the muscles. Without these neurons, the patient can't breathe, can't walk, can't swallow, talk. It's a relentlessly progressive, 100% fatal disease, and it will kill in typically 3 to 5 years following diagnosis. And the challenge that my project addresses is this. There is no treatment that even slows down this disease, much less stops it. What's my solution? My solution is to deliver to the motor neurons a gene that protects them from dying in ALS. A few years ago in my lab, we took one of the genes that was mutated in a rare genetic form of ALS and expressed it in a model organism. As we thought, that turned out to be toxic to the organism, just as it is to motor neurons. But because we were working with a simple system, we could do a large-scale experiment. We took 5,000 other normal genes and one by one put them into that system, looking for a gene whose expression would rescue the cells from the toxicity of the ALS protein. Out of all those 5,000 genes, only 4 were able to rescue the cell, and one of them did it better than the others. That gene was called UPF1. It was a normal human gene that is found in every cell in your body, and all we had to do was overexpress it by about 2-fold to rescue this model system from ALS toxicity. So our essential finding, which is what makes this research innovative, is that we found a therapeutic gene and we're able to deliver it to motor neurons, and when we do, miracles happen. We looked to see if exactly the same rescue could be achieved in actual motor neurons that were suffering from the same mutated ALS gene. Indeed, UPF1 expression was able to rescue motor neurons from the toxicity of any mutated ALS gene, and it also could rescue animal models of ALS. That, as you might imagine, got our attention. We're almost at the stage of doing the first human clinical trials of our UPF1 gene therapy for ALS. We first have to show that the virus, which we're using to deliver the UPF1 gene into the motor neurons, is safe, and that the route of administration we will use gets that gene to as many motor neurons as possible. We're doing those experiments now. But imagine what this will mean if it works to the 40,000 people in the United States with ALS and to their families. Well, that's easy. Is it safe? I sound like Laurence Olivier in Marathon Man. And will it work? By safe, I mean, will any side effects be tolerable? And by will it work, I mean, will it at least slow the disease down? Or better still, will it stop it cold?